Meersen is a preclinical research center dedicated to the search and development of innovative therapies against Parkinson, Huntington and Alzheimer's disease. In this context, Meersen offers a complete translational continuum of activities from in cellular models to various animal models, including non-human primates and up to phase 1-2 clinical trials. Meersen regroups on one site at Fontenay-aux-Roses all competencies and platforms in cell, gene and drug-based therapies necessary to perform preclinical proof of concept, including viral vector production labs, motor and cognitive suites, PET imaging and high-field MRI, all operating under BCL 1, 2 and 3 conditions. In a way very similar to previous collaboration with industry, and thanks to a large combination of preclinical platforms, Meersen can bring your molecule or your product of interest from the very first stage of development, for example in cellular proof of concept, up to a preclinical proof of concept in large animal models, and through its affiliated clinical centers up to a phase 1-2 trial. This can also be highly facilitated by interaction with the National Infrastructure of Translational Neuroscience, Neuratrice, Meersen being its coordinating center.
CEA offers cutting-edge platforms in health technologies, including multi-omics, computing, and medical imaging. The objective of the Passerelle project is the medical translation of the technologies developed in our labs. To do this, Passerelle connects academic labs and hospitals in partnership with private companies. Passerelle Imaging is a part of this ambitious program that focuses on imaging technologies and pharmaceutical companies. Here, the objective is to make our cutting-edge imaging platforms available for pharmaceutical companies. The idea is to use advanced imaging techniques in order to accelerate and de-risk drug development. Microdosing, or phase 0, as presented by Nicolas Tournier, are an example of these techniques. The Passerelle project is subsidized by 3 national research organizations, by Paris-Saclay University and by Paris Region.
Hello everyone, my name is Roger Legrand. I am head of the IDME department, which is a research department of the Jacob Institute of Fundamental Research Division of CEA, which is also a joint research structure of INSERM and Paris-Saclay University. The ambition of the department is to innovate in treatment and prevention of human diseases resulting from infection, immunological and hematological disorders. We have an exceptional set of assets in support of this ambition. We have a strong long-term expertise in studying basic mechanisms of these diseases. We have unique capacities in France and Europe for preclinical research, including non-human primate models of human infections. Basing our research at CEA represents a strong opportunity to support our research with the development of highly advanced technologies, including multi-omics, in vivo imaging, and for bioproduction. Finally, part of our teams and facilities are located at Bicêtre University Hospital, which is critical for access to patients and develop translational medicine programs. Because our unique tools, particularly for preclinical research, IDME department have been labeled as a national research infrastructure with a mission to offer our expertise, research capacities and technologies to academic and industrial partners in support of their vaccine or therapeutic developments. Today, we collaborate at the international level in the fields of HIV and AIDS, tuberculosis, influenza, pertussis, arboviruses, as a few examples. As an illustration of our contribution to national and international efforts, we have been significantly contributing to the COVID-19 pandemics by developing non-human primate models of this disease, allowing to test treatments and vaccines, some of which are today submitted to EU and US regulatory agencies for marketing authorization.
The CEA benefits from an expertise in radiolabeling a large variety of compounds, including small molecules and biologics, with positron-emitting isotopes. It is now possible to perform PET imaging with a larger and larger portfolio of compounds for use in animals and humans. Our team is dedicated to pharmacokinetic PET imaging. We use radiolabeled analog of new marketed drugs or new chemical entities with the same exact structure to study their distribution to target vulnerable and elimination organs in vivo. This is a fully translational approach which can be easily transferred and quickly transferred to humans. This corresponds to the concept of phase 0 PET microdosing. PET imaging is extremely sensitive and quantitative. Clinical biodistribution data can be obtained after injection of microdoses of the investigated compound. We think that human is the best model for humans. Several examples of clinical studies performed at CEA suggest that pharmacokinetic PET imaging can be performed in healthy volunteers or patients even before or parallel to the traditional phase 1.